In this session, we'll be discussing about third sexual reproduction method in bacteria, which is transduction. Now, transduction is an experiment which is done using bacteriophage. A bacteriophage is a virus which infects one bacteria. So, in transduction, what happens is the virus is used to transfer the genetic material from one bacterium to another bacterium. The experiment was given by Zinder and Lederbach. They worked upon Salmonella. You must have heard about Salmonella typhimurium, a typhoid-causing bacteria. Now, what happens? The virus infects bacteria in two ways. There are two ways. So, in first way, the virus will come. There are tail fibers. If you can look, there are tail fibers of the virus. With the help of tail fiber, the bacteriophage catches hold of the wall of the bacteria. And once it is what you call holding the wall of the bacteria, the DNA of that particular virus will be transferred to the bacteria. Now, it damages the bacterial DNA into fragments. Now, bacteria was using some machinery, some energy for duplicating the DNA, for forming the RNA, for forming the protein. Now, virus DNA has started using the same machinery because there is no bacterial DNA. The bacterial DNA are there in the fragments. So, virus will what you call multiply inside the bacteria. The virus DNA will be forming, virus RNA will be forming, virus protein will be forming, and eventually they will form what you call something like that virus, and the virus will be coming out, and this is how the lysis of bacteria is taking place. It is called lytic cycle of what you call bacteriophage infection. What happens in next? The virus is again will be transferring the DNA to the bacteria, but the DNA of the what you call virus will not be breaking the DNA of the bacteria. What happens is the DNA of the virus will be sitting along with the DNA of the bacteria. When it will be sitting along with the DNA of bacteria, each time the DNA of the bacteria will be replicating. The fragment those have been sent by the virus will also be duplicating. So virus must have come forward by taking some of the what you call DNA from any other bacteria. So now that DNA is being been given to this bacteria. So the bacterial recombination took place, which is transduction. Now, there are two types of transduction we see. There is one generalized transduction and one is a specialized or restricted transduction. In generalized transduction, the bacteriophage can pick up any portion of the host genome. It can pick any host portion's genome. But in a specialized transduction, the bacteriophage pick up only a specific portion of host DNA. If it is taking a specific portion, it will be restricted or specialized transduction. If it is putting any of the random portion, it is called generalized transduction. In next slides, in next session, we'll be discussing about bacteria related to human affairs.